Have you ever wondered why oxygen is released more in exercising muscle than in resting tissue, even though the blood oxygen level looks almost the same? Or why patients with fever or acidosis extract oxygen differently? The answer lies in a simple but powerful concept: the oxygen hemoglobin dissociation curve. In the next few minutes, we'll understand not just the shape of the curve, but why it shifts, what it means clinically, and how to think about it in exams. Let's break it down step by step. We'll first understand what the curve represents. Then we'll analyze its sigmoid shape. After that, we'll learn about P50, a very high-yield exam concept. Then we'll discuss right shift and left shift, including causes and mechanisms. And finally, we'll connect everything to real clinical situations. On the x-axis, we plot partial pressure of oxygen, PO2. On the y-axis, we plot percentage saturation of hemoglobin. As PO2 increases, hemoglobin saturation increases. But notice the curve is not linear. It is sigmoid, or S-shaped. Why? Because oxygen binding to hemoglobin is cooperative. When one oxygen molecule binds, hemoglobin's affinity for the next oxygen increases. This is called positive cooperativity. That's why the middle part of the curve is steep. In the lungs, PO2 is about 100 millimeters of mercury. Here, hemoglobin is almost fully saturated, around 97%. In resting tissues, PO2 is around 40 millimeters of mercury. Saturation drops to about 75%. That 22% difference represents oxygen delivered to tissues at rest. Now, imagine during exercise, tissue PO2 falls even further. The steep part of the curve allows rapid oxygen unloading. This is a beautiful physiological design. P50 is the partial pressure of oxygen at which hemoglobin is 50% saturated. Normally, it is about 26 to 27 millimeters of mercury. If P50 increases, that means hemoglobin requires higher oxygen pressure to reach 50% saturation. In other words, affinity has decreased. If P50 decreases, affinity increases. This is extremely important for exams. Always link P50 to affinity. A right shift means hemoglobin has lower affinity for oxygen, so oxygen is released more easily to tissues. What causes right shift? Increased carbon dioxide, increased hydrogen ions, meaning acidosis, increased temperature, and increased 2,3 BPG. Think about exercise. During exercise, CO2 rises, temperature rises, lactic acid increases. All of these shift the curve to the right. So oxygen is delivered more efficiently. This is physiologically intelligent. A left shift means increased affinity. Hemoglobin holds onto oxygen more tightly. Causes include alkalosis, low CO2, low temperature, and decreased 2,3 BPG. Fetal hemoglobin also causes left shift. Why? Because the fetus must extract oxygen from maternal blood. Carbon monoxide is dangerous because it not only reduces oxygen binding sites, it also shifts the curve left. So tissues become hypoxic even if saturation looks normal. Very important clinical point. The Bohr effect describes how increased carbon dioxide or hydrogen ions reduce hemoglobin's affinity for oxygen. This shifts the curve to the right. So in metabolically active tissues, where CO2 is high and pH is low, oxygen unloading increases. This ensures oxygen goes where it is most needed. Let's summarize. The oxygen hemoglobin dissociation curve is sigmoid because of cooperativity. P50 tells us about affinity. Right shift decreases affinity and improves oxygen delivery. Left shift increases affinity and can impair oxygen release. And the Bohr effect explains how CO2 regulates oxygen unloading. If you understand this concept clearly, many clinical scenarios become easy. If this explanation made the oxygen hemoglobin curve finally click for you, make sure to subscribe. We're building physiology step by step, clear, logical, and exam-focused. In the next video, we'll tackle acid-base disorders in a way that actually makes sense. See you there.